Welcome to the Explainer. Today, we're going to pop the hood on our very own cells to really understand the genetic engine that drives cancer. We're talking about the accelerators that get stuck and the brakes that completely fail. So let's get right into it.
So to really get a handle on this, let's think of a single cell in your body like a car. It's got these super complex systems, right? One says go, the other says stop. And cancer, well, you can think of it as a horrifying multi-car pileup, but happening at a molecular level. It's what happens when those fundamental controls just completely fail.
All right, first things first, let's talk about that accelerator pedal. You see, every single one of our cells has genes that are basically in charge of telling it when to grow and divide. We call these proto-oncogenes, and they're totally normal, actually essential for life. But what happens when that pedal gets jammed to the floor?
And this is where it gets really interesting. A normal proto-oncogene, that's your accelerator, but it can take just one single mutation, what scientists call a gain-of-function mutation, to turn it into a bad guy, an oncogene. And that's it. The accelerator is now stuck down, just forcing that cell to divide and divide and divide, completely out of control.
Okay, so how does this whole thing work when everything's normal? Well, it all starts with a signal from outside the cell. Think of a growth factor like the key to your car. It has to fit into the ignition, in this case, a receptor on the cell's surface. Once that key turns, boom, it kicks off this whole chain reaction, a pathway called the Ras-MAPK signaling pathway.
But how does that signal actually get inside the cell? Great question. Once that receptor is activated, it pokes a really critical protein on the inside called Ras. Now, Ras is basically a molecular light switch. It gets supercharged when it swaps one molecule, GDP, for another one, GTP. And that action flips the switch to the on position.
And once that Ras switches on, it's like tipping over the first domino in a huge, complex line. The signal just cascades from Ras through a whole series of other proteins, one after the other, all the way to the cell's command center, the nucleus. And what happens there? It tells these things called transcription factors to flip the switch on all the genes that drive cell division.
Okay, so that's the normal process. But where can it go wrong? Well, the first potential failure point is right at the beginning, the signal itself. I mean, what if the cell just decides, you know what, I'm going to make my own go signals? It's basically hot wiring its own ignition.
And believe it or not, that's exactly what some oncogenes do. Take VCIS, which comes from a virus, or a human gene fusion like CL1A1 PDGFB. These things can literally force the cell to churn out its own growth factors 24/7. It's creating this permanent go signal from the inside out.
All right, so that's one way. But what if the key is fine, the signal's normal, but the ignition itself, the receptor protein, is broken? Yep, that's our second major failure point. The receptor can get stuck in the on position, or the cell can just build way too many of them.
And we see this happen in a couple of nasty ways. You've got oncogenes like VERB that create a receptor that is just always on. It doesn't even need a signal anymore. Then there are others, like the famous ERB2 gene, a big player in breast cancer. It causes the cell to make so many receptors that it becomes hypersensitive to even the tiniest whisper of a go signal. And it's not just that pathway. Others like VMLP create a mutant receptor that messes with a whole different system, the JAK-STAT pathway.
So we've talked about the signal and the receiver. Let's move down the line to that next critical link in the chain, that molecular switch we mentioned, the Ras protein. And folks, this is one of the most common, most notorious failure points in all of cancer. Mutations can jam this little switch permanently in the on position.
Get this, mutations in the Ras family of genes, that's HRS, KRS, and NRS, are the most common human oncogenes we know of. They're found in a staggering 30% of all human cancers. Think about that. And what these mutations do is create a version of the Ras protein that has lost its off switch. It literally can't turn itself off, so it just keeps screaming that divide signal down the chain, nonstop.
But the chain of failure doesn't have to start at the top. You could have a perfect signal, a normal receptor, and a totally fine Ras protein, and things can still go wrong. The problem can be with the relay proteins further down the line, these things called kinase. They can become oncogenic too.
A classic example here is the BRAF oncogene. He makes a super hyperactive kinase, and it's a huge problem in many melanomas. And it's not just BRAF. You have other non-receptor tyrosine kinases like SRC, JAK, and ABL that can also go rogue. But at the end of the day, the result is always the same. That signal cascade just keeps firing when it absolutely shouldn't be.
And finally, we arrive at the last failure point. The problem can be right in the command center itself, the nucleus. The transcription factors that give the final go order can go rogue. Or, and this is really sinister, the proteins that are supposed to tell a damaged cell to self-destruct can be disabled.
And the troublemakers here have a few different strategies. You've got genes like the MYC family, MYC, MYCN, and MYCL. These are like master switches that turn on a whole library of growth genes at once. Then you have others like BCL2 and MDM2, which are, frankly, pretty insidious. They block the cell's own self-destruct program, a process called apoptosis. And if that wasn't enough, you have others like CDK4 and CYCD1 that just directly hotwire the cell cycle machinery itself. Okay, so that's the whole story of the stuck accelerator. It's a huge part of the problem. But think about it. Even a car with a jammed accelerator still has brakes, right? For a truly catastrophic crash to happen, those brakes have to fail too. And in our cells, the brakes are a class of genes called tumor suppressor genes.
How we even found out about these genes is just a fascinating story. Early on, scientists did this incredible experiment. They took a cancer cell and literally fused it together with a normal, healthy cell. And you know what happened? The new hybrid cell behaved normally. It was amazing. This was the proof that the normal cell must contain some kind of suppressor factors that could slam the brakes on the cancer.
And this brings us to a really crucial difference. Remember how oncogenes are a gain-of-function problem, a stuck accelerator? Well, tumor suppressors are the opposite. They cause cancer through a loss of function. We all get 2 copies of these brake genes, one from each parent. And for the car to crash, you usually need to lose both copies. It's not enough to cut one brake line. You have to cut both. So the big question is, how does a cell lose that last good copy? There's a process for this, and it's called loss of heterozygosity. It sounds complicated, but the idea is simple. Imagine a cell already has one bad mutated copy of a brake gene. Then, during cell division, there's just one little slip-up, and the one remaining good copy gets lost. One of the new daughter cells is now left with no functional brakes at all. And now, it's free to divide without any restraint.
Now these genetic brakes, they're not all created equal. You can really think of them in 2 big groups. You've got the ones that are like the actual brake pedal, the ones that stop the car. And then you've got the ones that are like the mechanic, the ones who do regular maintenance to make sure the whole braking system stays in good shape.
In genetics, we call these gatekeepers and caretakers. The gatekeepers, like the famous RB and P53 genes, are the foot brakes. They can directly slam the brakes on the cell cycle or even tell a damaged cell to self-destruct. The caretakers, on the other hand, are the mechanics. Their job is to maintain the integrity of the whole genome, repairing DNA damage before it can cause mutations in other critical genes, like the gatekeepers themselves.
And here you can see a great list of some of these critical players. On the gatekeeper side, you've got your heavy hitters like APC, RB, and P53. These are the direct stop signals. Then on the caretaker side, you have genes that you've probably even heard of, like BRCA1 and BRCA2. Mutations in these genes are so famous because when they fail, the cell's entire DNA repair shop is crippled, which dramatically increases the risk of cancers, especially breast and ovarian cancer.
All right, we've looked under the hood at the accelerator, and we've checked the brakes. So let's bring this all together. Why does knowing about all these specific broken parts matter so much when it comes to actually fighting cancer?
Because at the end of the day, that catastrophic crash we call cancer is almost always the result of a double whammy. You've got a stuck accelerator, an oncogene, and you've got failed brakes, a lost tumor suppressor. It's that combination that's so deadly.
And this is what leads us to really the most powerful idea in modern cancer therapy. If we can figure out the exact parts that are broken for each person's cancer, can we build the perfect personalized repair kit? By identifying the specific oncogenes and tumor suppressors that have gone wrong in a patient's tumor, we can design therapies that target those specific failures. It's all about moving away from just carpet bombing the system and toward a future of building a precise, personalized repair plan for this incredibly complex machine.